Okay, so in this video, I'm going to be going over the remaining parts of chapter 14, which is going to be on these patterns of inheritance. So the previous video was about probability and kind of basic dihybrid crosses, how to solve for them. I have another video that I'm posting as well where I kind of walk through different versions of genetics problems. This one is going to be more about kind of the theory of what's going on with each one of these. So make sure that you conceptually understand what's going on. So let's kind of just get going right off the bat off of what is an example of codominance. And codominance is, as the name implies, where both alleles are going to show up. There's no recessive version of it. So basically each one is going to be expressed equally. So the example that I've used is Tay-Sachs disease as well as blood typing. So basically, this is an interesting version where if you're homozygous for the disease, you have the disease. You could be heterozygous and you have the disease without symptoms, or if you have the no disease trait, they're both expressed. So that's kind of a weird one. The more common one is going to be blood typing, where there's multiple versions of an allele. So basically, you can have type A blood being homozygous carrying the A, and homozygous, or I should say carrying the A and then also carrying the O. Whereas if you're type B, you could be homozygous for B, but also heterozygous for B. But you can actually, it's this one where they're both going to be expressed. The A and the B antigens are both going to be expressed. So I'll have a more detailed version of how to solve blood type in just a second, but basically just both traits would be showing up. So let me kind of walk you through this version on multiple alleles. So blood typing is an example of having multiple alleles. So you can have type A, you can have type B, you can have type O. And these things on the surface are antigens. And these antigens are basically proteins that your blood cells have. And we didn't cover the immune system yet, but basically your immune system, if you are type A, all of your red blood cells have a specific protein on the surface. And the reason why that's important is because let's say if you are type A, but you accidentally get some type B blood in you, well, your body recognizes those antigens as foreign, and it'll basically break it down and recognize it. So let's talk about it, where if the allele representation is a little bit interesting, where if you represent it like this, that means you're actually going to be expressing the A antigen. If you carry the B, you're going to be making the B antigen, and both of these are codominant, as well as multiple versions or multiple alleles, where lowercase I represents type O. And basically all this is saying is that A and B are both dominant over the I allele. So what I mean by that is if you look at little I, if that's your particular genotype, notice that you don't make any of the antigens. Now, this is that example as well of being codominant. If you have IA, IB, A and B are both expressed. So this individual, if you are type AB, if that's your blood type, you have both the A antigen as well as the B antigen. You actually express both because both are dominant. Now, looking at this though, let's say if you're type B and this is your genotype where you actually have the allele for B, but you also are carrying the O allele, it doesn't matter because the B is going to be expressed over the O. So don't necessarily worry about the antibodies right now in this video. This is just all about doing the genetics. So this slide is talking about having multiple alleles. You can have the A allele, the B allele, and the O. It's not just like dominant or recessive. There's actually more than one for the gene. So let's kind of do this one. If a woman has type A blood and a child has type O, list the following. You got to figure out what are the blood types that the dad could be and what are some blood types that the dad could not be. So let's talk about the woman first. So if we're looking at the woman, her possible genotype is going to be a couple of things. She could either be this, she could be homozygous for A, or she could also be this genotype, right? And because if you took a blood sample of the mom, we don't look at their genotype, we just look at the phenotype, which would be type A. So mom's blood type, she would have that kind of particular blood, those antigens. So her genotype is either this, or it's going to be this, okay? Now her child is going to be type O. So if the child has type O, you know that his or her genotype is I, okay? Now, right away, this should kind of change your opinion on what mom's blood type is going to be. If mom's genotype was homozygous dominant in the Punnett square, there's no chance that the baby could be type O. So I now know that mom's genotype must be heterozygous because there's no chance that the baby wouldn't have gotten one of the dominant A's when you do the Punnett square. So let's walk you through how to do a Punnett square with the blood types. It's pretty simple. So we just got to figure out a couple of things. And I'm going to go ahead and put mom's genotype up here. So she would be, okay, she's heterozygous. So let's tackle the first one. What are the blood types that dad could be? Well, let's see if dad is going to be, could he be type A? Well, let's say he has the same genotype as the mom in the sense that he's heterozygous. So if I did a Punnett square like this, it would look something like this, where there's a chance the baby could be type A. This baby would be type A. This baby would be type A. But right here, is there a chance that a dad with type A blood could be the father? Yes, so dad could be type A. Now, what if I do it again, but I'm going to change dad's blood type. I'm going to erase all this. So dad could be type A. So if he was type A, he would still be at, he could definitely still be the father. But let's set it up again. And what if the dad claims to not be the father based off of his blood type as being B? So let's kind of take a stab at this one. So what if dad is type B? All right. So if dad's type B, we can kind of assume the same thing. I'm not going to do the entire Punnett square, but if he's type B, again, we're making the assumption that he's heterozygous, that he could still be carrying that O allele. So you can see right away that he absolutely could still be the dad. All right. Now, if he's type B, both of those, type A, type B, that could be dad's blood type. What about if dad was type AB? Is there a chance that he could be the father if he is type AB? Well, let's go back and I'll erase this and do the Punnett square here. So I'm going to keep mom's blood type as being A. That's mom up there. I'm not changing that one. But what if dad is AB? Well, if dad is type AB, this is his genotype. And we can do the Punnett square cross, where if I crossed all of these, that would be a child that has type A. I would get a child that would be AB. I would get another child over here that would be type A. And this child over here would be type B. So this kind of answers the second one. If dad was type AB, again, because it's codominant, he's not carrying the O allele, dad could not be AB. He couldn't be type AB because there's no chance that they could produce an O blood type child. So could the dad be type O? And the answer would be yes. We can go through and do that Punnett square as well. So just to kind of put this all together to wrap it up. So I'm going to go ahead and eliminate that he couldn't be AB, but what if he was type O? And again, that would be his genotype. Okay, so if mom's type A, I think you guys can see this right away. At least I hope you can, that if he was little I, and I cross it with another little I, yeah, you're going to get over 50% chance that your baby is going to have type O blood. So this would be a 50% chance of having A if dad is type O. So he is definitely, he could be type O, type A, type B, and still produce a child with the mom and give it type O. He just couldn't be AB. So dad could not be AB because of that one Punnett square. So that's an example of a couple things. That's an example of having multiple alleles. We have the A, the B, and the O alleles. And it's also an example of codominance in the sense that A and B are both expressed dominantly. Okay. Now, Let's get into this one. This just explains another thing about incomplete dominance. And that's basically where the traits are going to be blended. So when you look at it phenotypically, it's kind of a blending of the phenotypes. So basically, if this flower was this and this, all of these flowers would be this particular genotype. And you get that expression of like that pink color. Okay, now that would be an example of incomplete dominance. Now, what if it gave you a scenario where this was, let's say it was another problem, and just so you guys get an example, if this was co-dominant, so basically if it changed it and you got a question where it was talking about having a co-dominant trait, let's kind of explain what that one would look like. And that one would look like this. If they said that red and white are co-dominant, let's explain what that would look like. If you were if you were that genotype, it would be red. And if it was little w, and that was white. If you were this, the flower would have some red spots and some white spots, no kind of blending of the trait. So just be aware on if it tells you in the problem if it's co-dominant. Because incomplete is where the phenotypes kind of blend. Co-dominant is when they're both expressed equally. So this is showing you an example when it kind of gets that blended trait, it's going to be incomplete. Co-dominant is when they're both still evenly expressed. Okay. So I'm going to hold off on this one because I have another video where I go through each one. But this is just about the concepts. Now, plectropy is going to be pretty simple, and there's not a whole lot that I can basically emphasize more than this. Let's just, I'll highlight it. Let's say you had sickle cell anemia, right? That let's say this is the gene sequence that gets mutated, okay? So I'll put a little sad face there that is where the mutation is. Now, let's talk about traits. Maybe you would be tired. That's the trait. Maybe you would also necessarily be weak. And let's just say we'll do low muscle mass. And so what I'm trying to emphasize here is that let's say you did have that genetic disorder of sickle cell anemia. You have a faulty gene, but that one gene is going to be associated with multiple phenotypes or multiple traits. So that's what it's saying, is that that one gene can actually have multiple symptoms. So if you're looking to be a doctor or a nurse or anything in the medical field, oftentimes multiple diseases or multiple genetic disorders can have overlap of traits. So we got to kind of look at all of them. Now, if I had another genetic disorder, let me just try and highlight this at a different color. Let's say if I had another genetic disorder, let's say gene 2, well, that's only going to result in these two traits, so being tired and weak. So just understanding those, that's kind of the overlap. The different genes can have multiple symptoms with it. So that's what plectropy is standing for. Now, epistasis is a bit of a trip. It's kind of like an on-off switch. So I like to kind of always draw out this picture to explain epistasis. So give me just a moment while you're with me and understand how I'm drawing out this. So how I like to explain epistasis is we have really two genes. I have one gene that's going to determine, let's say, fur color expressed, and that's going to be a yes or no, kind of like an on-off switch. And then you have another gene that's going to be the actual color of the fur. So basically how I think of it, when I think of epistasis, it is literally like kind of an on-off switch. So you have this gene is actually going to control the expression of that gene. So think of like albinism. That's typically what this is going to stay. So when it says one gene alters another gene at a different location or a different locus. So basically what this is showing you is, according to this lab, it looks to be that big B is going to determine the color. So we're going to say that big B is representing, we'll say, black, right? And we're going to say that that's dominant. Little b is going to be brown, and that is actually going to be recessive. So it's the big B and the little b are determining the color. But what about the E? And that's where things get a little bit interesting. So if you notice that when you look over here, you're saying, well, I have this, I have a black lab, I have a brown lab, but then I have this golden lab. And that has to deal with the second gene. So let's think of it this way. It would be something along the lines like this. When you look at the genes that these guys have, sorry, I'm just trying to change the color. Let's take a look here. I'm going to write this over. So the big E is going to represent the color is expressed, meaning are we actually going to make the fur color or not? And little e means that no color is expressed, okay? So basically think of it this way. If this is my little diagram over here, if you were big E, or big E, little e, basically, according to this, this means that according to the dog's genotype, if he's big E or big E, little e, that means he's actually going to express color. Well, what color? Well, that's going to be determining here. If they're big B, little b, or little b, okay? So let's look here. Let's take a look at this dog. His genotype is this, okay? He's little b. That's what makes the dog brown, but he's also big E, which means that, yes, he has the ability to actually express that fur color. Now, I'm going to jump down here. Look at this dog. Well, this dog is little b also, but if you notice, this would actually be turned off because little e is the genotype he has. So the fact is he would be brown, but the fact he has that little e, which is recessive to not expressing anything. So that means it doesn't matter what that dog's genotype would be, you're not even expressing the color. So think of epistasis almost like a on-off switch. So you have one gene that determines, are we going to express these at all? So in this scenario, if you were a little e, it doesn't matter what the dog's genotype was, you're not going to express the color. So if the dog was big E, or heterozygous, big E, little e, then yeah, you're going to go ahead and express the color. So one final example, let's look at this dog. He's big B, so he should be black because black is dominant over the brown. And he also has the dominant gene to express fur color in the first place. So that's what epistasis is. Now, that is something that is important, but even more important to me would be understanding polygenic inheritance. So there's all kinds of different scenarios here where basically it means multiple genes affect one particular phenotype. Skin color, eye color, height, for example, works like this. Now, let's think of this. This will be my example here. So this kind of shows you a nice distribution. And I like to kind of explain it that each dominant allele is going to increase the darkness, and that's due to the pigments of the skin. Okay, so basically what I mean by this, if you look here, if these two individuals cross, this is showing you a distribution that if you look at this individual, right, big A, big B, little b, big C, little c, I have three dominant alleles. Now, if I compare that to a person that was big A, big B, little big C, little c, they have one more dominant gene or one more dominant chromosome. Now, this gets a little intense when you think about it, but the easiest way to understand it is that the more dominant, if you're doing something like polygenic inheritance, the more dominant alleles is going to be equal to an increase in protein synthesis. So really what I'm trying to emphasize here is that this person is making a little more pigment because they have one more dominant gene than this individual. So, and then even furthermore, if you have this person that had big A, big A, big B, big C, and little c, they have an additional dominant version, so they're making a little more protein. So that's really one thing that you got to understand when we think of polygenic inheritance. As the name implies, poly means many. It basically means many genes are going to be associated with one particular phenotype. The more dominant genes you have, the more of that particular protein you're actually going to be making. So if you think about it from like a genetic standpoint, and this makes a lot more sense that you covered protein synthesis. If this was a chromosome, all of these genes are going to help. I'll say gene 1, gene 2, gene 3. All of these are going to be expressed to help essentially make the same protein. In this case being pigment in the skin. Now, vice versa, if you go on the opposite end, if you don't have any of those dominant traits, you're not going to be making any of that pigment, any of that protein, hence why you're going to get a very fair skin. So that's what, when you get any type of phenotype that is quantitative in the sense like height, darkness, or lightness, anytime you get varied degrees of a characteristic, not just like tall, short, one particular phenotype versus the other. Anytime you get some variance and it's quantitative, typically what's going on, it's polygenic inheritance. Multiple genes are associated with one particular phenotype. The more dominant it is, the more it's actually expressed. So that's what polygenic inheritance would be. All right. Basically the last kind of couple ones that I need to kind of go over is how to read some pedigrees. And pedigrees are not difficult, but there are a few things that you guys need to make sure you feel comfortable with. So this is talking about the 1, the 2, the 3. All of these over here, these are talking about generations. So this is the first generation, second generation, the third. Anytime you get this cross, this is talking about that they mated. So you followed through, and the lines going down represent how many offspring they had. So if these two mated, hopefully you're able to understand that these people had 1, 2, 3, 4, 5 children. They'll give you a key like this, so don't necessarily let that freak you out. Squares are going to be males, circles are going to be females. Now, if you look at this individual, if I numbered these, 1, 2, 3, 4, and 5, hopefully you're able to understand that the first child in the second generation mated and had 2 kids. The second male son, the son mated and they had 3 kids. The third and fourth women on the second generation, they didn't have any kids. The fifth one, that daughter actually mated and had 4 kids. So they'll always kind of give you a scenario of how to read them, as well as you're just tracking an appearance or a trait over generation. So let me kind of give you an overview of how that would look like. So the genotype of individual 1, so basically we're trying to figure out what this individual's blood type would actually be. So we got to take a look at the offspring. So this is really 2 things. It's a pedigree as well as a genetic problem on blood typing. So let's figure out this person's genotype first. I'm going to look at individual 2. I got to do a quick glance at the kids. So that's what we're solving for here. Now, right away, I know that this person's genotype is going to be this. I'm going to make a huge understanding that you're able to follow with me on that, because the fact is that this person with type B blood, it produced a child with type B, it produced a child with type AB, but the big indicator that I know that this individual was carrying the O allele was because of this child number 3. Because the fact is, if he was homozygous for B, there's no chance that he could produce a child with type A. So I got to figure out what this person's blood type is. Now, we got a couple of options here. It would be a bad call for A, because if this person was type O and you made it with type B, there's no chance that you could get a child with type A. So that one's out. For that reason alone, I'm also going to take out C. I couldn't have 2 people with blood type of B, just B, giving a child of type B. Let's figure out why what's better, B or D in this case. So I'm going to go out on a limb and just say, okay, I think that if this person was heterozygous for A, there is a chance that a child could be AB. And again, you could do the Punnett square off to the side here. That if you were type A, let's see if all these genotypes match up. I could get an AB here. I could get a type A carrying the O allele. I could get a type B carrying the O allele. And I could also get a type O. So don't let this freak you out. Just because I got a type O in my Punnett square and I don't see one here, that's okay. The fact is I got a type B here. I got a type A right there. So I got a B, I got an A. I also have an AB. So I think I'm going to go ahead and select B for my answer, and that is going to be correct. The reason why it's probably not going to be homozygous for A is that there's no, let me double check this on the Punnett square, and we'll kind of see why it can't be that. So I'm going to keep this. This is showing you the dad's blood type. But let's do a cross and see if we can get into the same issue if both would be correct. So let's just see and figure out why the dad couldn't be that particular genotype. So, excuse me, not the dad, the mom. So I'm selecting it as B, but I'm just going to show you real quick why it couldn't be D. So if I cross it here, I'm thinking, okay, well, why can't it be D? Let's figure out why. Well, if I cross it here, this person would be AB, so that matches up. That could still be the case. If I crossed it here, I have a type A. That makes sense. Now, if I crossed it here, I would get another AB. And if I crossed it here, I would get another A. So there is no chance with that genotype that I could get this child with type B. So that's why B is a more correct. The answer choice B is better than the answer choice of D. So these were going to be the two genotypes that had to be correct. So number one, the answer choice for this one is B. Let's kind of knock this one out real quick. Use the phenotype chart, the pedigree, to answer the following questions. How many children did parents A and B have? So we got A and B here. We just got to follow through. They had 1, 2, 3, 4. So they had 4 children. Okay, indicate the genotypes of the parents. Give the genotypes of M and N. So let's figure this one out. I don't know if it's going to give us any pre-context to this, but it's telling you that this male is normal. So I'm just going to write that again, and I'm following the color coordination. The female is normal. Okay. Okay. Now here we go. Let's look at this male. He has. I'll just put the DIB for diabetic. So they had a child that was diabetic. They also had a female that was diabetic. I'll just put an N here for normal. They had a normal daughter. They also had a normal son. So let's figure this one out. I'm going to basically make a little chart off to the side. I'm going to say that capital D is going to be normal. And I'm going to say that that's dominant over lowercase d, which is going to be having diabetes. Okay. And I'm going to say that that is recessive. And here's why I kind of get this inclination that diabetes is a recessive condition. Because when I go back and double check it, and if I said that this is dad's genotype, that's mom's genotype, you would kind of do a Punnett square off to the side. This isn't any this weird kind of scenario where they'd get this 1 or 75% chance that their kids would be normal, and a 25% chance that they would have diabetes. Now, let me just double check this, that the parents, again, the mom was normal, the dad was normal, and both their sons and daughters, one son has diabetes, the other daughter has diabetes, but they also have the normal son. Now, And that's okay. I would say that diabetes is a dominant trait according to this. I'm seeing it equally in males and females. And that's the only real scenario that I would come up with in my head to make this make sense. There's no chance that just all of a sudden that trait shows up without there being like a random mutation. You would have to know that for this problem. So indicate the genotypes of the parents. They would have to essentially be heterozygous. Okay. Give the genotypes of M and N. Okay. Now let's look here. This female, she was diabetic, but look at the male she actually married. That male was also diabetic. So we have two individuals that are diabetic. We just determined having diabetes was a recessive trait. So they're little d, little d, little d, little d. So, and if you look at their one child, the evidence they're giving you is that that child has diabetes. These individuals would also have to be diabetic as well. So that's kind of one way to tackle these Punnett squares. So I'm going to go ahead and call it for this one. I think the other ones are going to be pretty, that's kind of just the ins and outs, the basics of the Mendelian genetics, as well as kind of, I don't want to call this quite yet advanced genetics, but just really pedigrees and some other interesting scenarios. We'll see you in the next video.